I think after Chetan's talk, it will be tough to compete. So I will give a I'll give a talk that is something new, something what I think India doesn't look at, and something which I learned during my postdoc as well. So today I'll be talking about unearthing the metabolic basis of angiogenesis using systems biology approaches. Okay. Before starting the talk, I want to introduce my department and also the themes that we work on, because our department is young. So I'm an assistant professor in the Department of Biotechnology, IIT Hyderabad. Our department was established in 2010, and we take lot of different, we offer a lot of different courses from BTech to PhD, and we have well-equipped laboratories for wet lab and dry lab research. So in this building, actually this part is biotechnology and this part is biomedical engineering, and we sit somewhere in between. So that's and if you look at the department from like the top angle, it has a shape of a chromosome, just for fun. Okay, our department has works on multiple different research themes, like structural biology, disease biology, cancer biology, big data biology, and bioprocess technology. What I want to highlight is that out of 15 faculties, 4 of them are core computational faculties, including me. So we work on computational problems. My research interests specifically are to understand the design principles of biological systems using different approaches, and I work at the molecular level where I try to understand the interactions between different molecules and thereby make some biological comments. My group is just 9 months old. I've just joined IIT, and I have currently recruited 4 PhD students, 2 master students. So we are still growing. If you guys are interested, you can contact me later. My current research interests include parasite microeukaryote metabolism, host immunometabolism, and molecular biomarker prediction in parasitic lung infections. Okay, let's come back to the talk. The sales pitch is done. So the topic is again understanding the metabolic basis of angiogenesis using systems biology approaches. So how do we go about it? So let's try to understand what angiogenesis is in the first place. So I'll show a video which is let's say 1 minute long, I think, maybe even lesser. What you can see in this picture is a tissue and a blood vessel passing through the tissue. And also what you can see is that each of these, the lumen of this blood vessel is lined by these cells called as endothelial cells. Those endothelial cells will be the focus of the topic. And what you can also see is that the tissue is actually producing a protein called as VEGF, vascular endothelial growth factor, that is going to induce the formation of a new blood vessel. So the process of angiogenesis actually forms new blood vessels. So I'll just show this video quickly. So what you can see is that the VEGF stimulus actually activates this endothelial cell to form a tip cell, which is purple in color, which has philopodia, which is followed by the stalk cells. And the sprout is actually moving towards the direction of VEGF. You can also see switching between different endothelial cells. So any stalk cell can become a tip cell or vice versa. Okay, this particular phenomenon helps the blood vessel sprout to move towards the direction of the VEGF stimulus. Now, when you have 2 sprouts coming from different directions moving towards the stimulus, they merge together to form a blood vessel. Okay. Now these stalk cells, basically, if you looked at the video carefully, the stalk cells are actually increasing in number, so they are proliferating, and these tip cells actually are elongating in size. There is only 1 tip cell per sprout. Okay, and so this is basically involved in migration. Okay, these endothelial cells in the end form this bunch of network of capillaries, arteries and veins, and they are involved in multiple pathophysiological roles. Like for example, you would see this in wound healing, during exercise, menstrual cycle, placental angiogenesis, embryonic angiogenesis, all of which requires new blood vessels for their function. From the pathological angle, what you can see is that you can also have impaired wound healing. You can still have aberrant angiogenesis. You can have hyperproliferation of these endothelial cells in tumors. You can have choroidal neovascularization where there is leaky blood vessel formation in the choroids. We'll talk about this more. And there is also atherosclerotic angiogenesis where the atherosclerotic plaques in the blood vessels induce angiogenesis. Okay, lot of information, but this is important to understand. Now, if we just look at a single sprout that we saw in the video before, okay, it consists of these stalk cells, as I told you, and these were proliferating, increasing in number, and you had the tip cells, which were just producing these philopodia-like things so that it can move towards the stimulus, right? So this particular tip cell actually requires ATP so that it can move towards the VEGF stimulus. And the stalk cells, of course, they are increasing in number, so the biomass has to increase. And also you can see that there are some extracellular matrix components that will actually provide a structure to the sprout so that it can move in this direction. So looking at this image, you might think I am doing some physics, understanding the dynamics. No. What I want to look at is how this, the different pathways, metabolic pathways involved in these different functions of migration and proliferation. Okay, so that we can understand angiogenesis holistically. So our basic biochemistry textbook says metabolism is a totality of all chemical reactions, and you have catabolic reactions and anabolic reactions. Catabolic reactions actually break the nutrients to get energy. Anabolic reaction use energy to build essential cell components, right? So let's connect that to angiogenesis. This particular slide, I want to focus on how this, if we are able to understand metabolism in angiogenesis, how can we kind of target the pathological situations. So in general, if you look at any angiogenic targeting paper, they will talk about they'll talk about targeting this VEGF stimulus because that is inducing angiogenesis, right? So if you are able to target the driver, no more the engine is going to run. But this is the conventional anti-angiogenic therapies that are available. They are even available for cancer. But the problem with this approach is that if you block VEGF, you will have other growth factors that take over, and thereby angiogenesis will still continue, the pathological angiogenesis, and there will be therapeutic resistance. So my postdoc lab where I did this work, actually their hypothesis was that why not target the metabolic engine, because ultimately we know that metabolism is required for proliferation and migration, and if you are able to target the engine itself, it doesn't matter which growth factor is trying to activate the particular blood vessel formation in a particular situation. So easiest way was that focus on metabolic pathways. So if you look at literature regarding this angiogenesis, what you see is that, like for example, this migration phenomenon which I talked about, you see that glycolysis is one of the most important pathways, and 85% of ATP that is required for movement of this tip cell is produced through glycolysis. What you also see is that the stalk cells actually use a different pathway, and not for ATP production. The aim is to optimize biomass, as I told in the first few slides, right? And what they see is that a novel pathway, fatty acid oxidation, is actually used for DNA production. If you know your basic biochemistry, DNA is produced through using amino acids, right? But this fatty acids being used for producing DNA is something very specific to endothelial cells and maybe one more cell type, I think mast cells. But otherwise, no other cell types use fatty acids to produce DNA. Apart from that, the electron transport chain works not only to maintain redox balance, but to produce amino acids. Again, this is an interesting phenomenon which is specific to endothelial cells. Okay, there are other pathways that are also known in cancer, like serine biosynthesis and glutamine oxidation. They have very similar function in endothelial cells. Okay, now why am I telling this history? Because it's important to know that metabolic pathways do not work in isolation. If those pathways have to work, then there will be some alternative pathways that have to couple with those existing pathways so that you can have better function, okay, or an optimal function. So to actually understand this, what we can do is you can just look at the purine and the pyrimidine rings and find out what are the sources that can produce carbons or nitrogens in this ring. And you will understand that, like for example, as I was telling, that glycine, aspartic acid, glutamine, these are certain amino acids that will be used to make this backbone, okay, for both the purine and the pyrimidine rings. Now, these amino acids, if you know your basic biochemistry, it will come from the TCA cycle. So potentially, there can be any metabolite that can enter into the TCA cycle so as to produce these amino acids. So this is very interesting because you have a flexible metabolism now, and it's very tough to understand which pathways are going to be chosen in this case. Also, if you just look at biomass generation, biomass is nothing but lipids, proteins, and nucleotides. If those have to be produced using some input nutrients by the cell, actually they have to, all these have to be produced simultaneously. This kind of is helped by this redox factors. So basically reactions, some reactions are oxidation specific and some are reduction specific, and both these reactions have to couple with each other so that there is a redox balance, and thereby achieving the redox balance will also optimize the production of these different metabolites. Okay, lot of facts, right? But it's important to understand that metabolism is complex, and just knowledge of a few metabolic pathways will not give you the full understanding. Okay. So if you look at biomass, okay, as I told you, biomass is made of proteins, DNA, RNA, lipids. Now, proteins are made of amino acids. You have DNA that is made of these deoxyribonucleotides. You have RNA, which is made of the ribonucleotides. You have lipids with like sphingolipids, phospholipids, ceramides, so on and so forth. So if I just want to find out which pathways are synthesizing these, I will end up with this huge picture of different reactions that can make those metabolites. Now, how do I really look into this map and find out, okay, which is the best possible pathway that can produce all those different metabolites in the output, right? Furthermore, it's the same metabolic network that is kind of being used by different cells in the human body, right? So how do I find out, let's say for example in this particular condition, which of these pathways are going to produce biomass or ATP? How do I come up with this given these input conditions? It's not a very easy task, right? So ultimately the question is, how am I going to find these catabolic anabolic routes that can kind of optimize angiogenesis, right? So ultimate questions what we ask in our work is, are there any unidentified metabolic reactions that couple to these existing pathways, and can we use some systems biology approaches that can deal with these big systems? Okay. So I will kind of highlight these questions with respect to one specific problem that we did during our PhD. So we were working on this wet age-related macular degeneration. So in this particular condition, what happens is that, let's say in aged people who start losing vision, what happens is that you have these leaky blood vessels in the choroid, and these leaky blood vessels actually proliferate profusely. And because they are leaky, the nutrients and proteins, they start accumulating in the choroid, thereby affecting the vision. So what happens is that it's almost, apart from VEGF, anti-VEGF therapies, there is no other therapy that is available for these patients. So we wanted to find out whether we can understand the metabolism and thereby provide some targets for this particular disease. Okay. My colleagues were actually testing this hypothesis in preclinical mouse models of age-related macular degeneration, and they were using a particular technique called choroidal neovascularization. Basically, what that model was is that you take mice and you laser burn the eyes, and then you pop out the eyes, dissect them, and look under the microscope. Okay, is there any change in the morphology of the blood vessels? What the problem with their approach was that they were only looking at the morphology. How is that going to help with understanding the molecular details? So we came up with a plan that maybe we should look at the single cell level changes that's happening within the choroids. Because it's possible that these novel endothelial cell populations which are emerging, that actually leads to these leaky blood vessel formation. And if we are able to find out that progenitor, maybe then we can able to kind of target them. So what we did was, basically we took mice which were not injured by laser, and those were our control, and rest of the mice were choroidal neovascularized, and we tried to compare these two scenarios. Okay, what we found out is that we obtained around, let's say, 10,000 healthy single cell choroid endothelial cells and some 18,000 laser burned choroid endothelial cells. You can already see that the numbers of these lasered choroid endothelial cells, they are actually high. Okay, which means there is some increase already in the number of endothelial cells. Okay, so what basically is this single cell profiling? It's like, you can understand by this analogy. This is a very popular analogy that is given for single cell RNA sequencing in any talk actually. So like, you can assume that this organ or tissue is like a bunch of fruits that that's kind of liking each other, working with each other. And if you just blend the tissue, you will get a smoothie. And then you are going to just do some analysis on the smoothie, right? That smoothie actually is just a total amount of RNA that is present in your tissue, for example. Okay, so that particular RNA sequencing methodology is bulk RNA sequencing, where you are not concerned about which RNA is coming from which particular cell type. Rather, if you can look at single cell RNA sequencing, where you can characterize which RNA comes from which particular cell type and how much is it available in each of the cells. So we used, this is a commercially available sequencing pipeline. So basically what this experiment actually does is that you digest your cells, you provide it to the flow cytometer, you have let's say some gel beads to which these digested cells actually they bind to those beads. Then later on you encapsulate them in a oil emulsion, and each of these oil emulsions actually contain one single cell. And interestingly, while doing this, they also ensure that each oil emulsion has the components of doing enzymes that are required to perform polymerase chain reaction. Okay, so that you can quantify the number of RNAs that are there within a single cell. Okay. Apart from that, because there is a barcoding feature, you can get which cell the RNA is coming from, and also how much, which gene is it associated with, and which cell it is coming from. So both informations you get at one go. So what we do is, once we have these reads, we can do number of processing steps so that we end up with the transcriptome profile of every single cell, and thereby doing simple clustering techniques. It's not actually simple, but simple clustering techniques, you can get these structures of RNAs of cells that are similar to each other. So for example, in these two plots, these are TSNE plots, T stochastic neighborhood embedding plots. I'm not going to tell what exactly it is. It is used for visualization purposes of these different clusters. And what you can see is that each of the dot actually represents a single cell. Okay. What we did was, we plotted the two things separately, and we understood that there is a particular cluster of cells that specifically occur in this CNV situation, where the laser burn is given to the eyes. Okay, so this was interesting because you have an injury specific cluster, and that is, as I showed in the previous slide, the CNV could be a good model for AMD, understanding age-related macular degeneration. So we need to focus on that cluster. So what we did was, we did some differential expression analysis. Basically, we are trying to compare every cluster with every other cluster and getting some genes that are upregulated in every cluster. Okay, upregulated meaning having higher expression. And based on this higher expression, we are trying to mark each of the cells as a particular cell type. Okay, thereby we mark these different cells, And based on this, we understood that this injury cluster actually has all the components of an angiogenic function. Like for example, you have proliferating cells, tip cells, you have some transitioning cells, some immature cells, probably in the phase of making the proliferating cells. So all of this together kind of gave us an understanding which cell types are present in these leaky blood vessels. We also compared these two situations, okay, and did some gene set enrichment analysis just to understand which pathways are changing transcriptionally, and we can see that biomass and ATP synthesis genes, they are actually changing, okay. And I mean here there is actually a misprint. This should be proliferation and migration, because in our previous slides we saw it, ATP and biomass synthesis, they are two different processes occurring for both tip cells and stalk cells, okay. Okay, this is all fine, good, but a transcriptional change may not lead to a metabolic change, okay, because people have seen in multiple papers that the correlation between transcription and metabolism is very poor, although it can indicate some things, right? So we thought, why not we take the map, we make a genome scale metabolic model. You guys must be aware of this. It was discussed in the previous talks also. So the map can be taken and some conditions can be added. These conditions come from basic biochemistry, like stoichiometry, mass balance, reversibility, and occurrence of these different reactions in different subcellular compartments. We combine it with the uptake rates of different metabolites, and which can be estimated from media composition and the cell number that you have. And in the end, we can use it for optimizing certain biological functions like biomass, ATP synthesis. This is what we wanted to understand. We wanted to understand which pathways are actually contributing to these metabolic tasks, okay? So we perform a very simple analysis. We do flux balance analysis. So what FBA exactly is that, let's say you have this linear pathway and the aim is to maximize this, let's say glyceraldehyde 3-phosphate production, okay, and you have glucose as the input. Now what we are trying to do is we want to estimate the flux values of each of these reactions, okay. Now the only inputs that we have are the uptake secretion rates and the map, okay, the map that we saw before, and or let's say this simple map here, this linear pathway, okay. What we can do is we can write equations for the consumption and production of every metabolite. So if you see, let's say for example, this particular reaction, this extracellular glucose, it is produced by rate V1 and actually consumed by this rate V2. So in the end, we'll have linear looking equations actually here, okay. But the problem with this approach is that it is not scalable for the entire set of reactions that are occurring within the cell. It's good for maybe this particular small pathway. So how do we work in that case? So what people have devised is that they can decompose this matrix into the product of these two matrices, stoichiometric matrix and the flux vector, assuming that this entire system is working at steady state, okay, because metabolic reactions are typically faster than, let's say, signaling or transcriptional changes. People assume that this steady state, 5 more minutes? Oh my God, okay. I'll then quickly rush through. Okay, so ultimately what happens is that you end up solving this system of equations assuming an objective function, and thereby you want to estimate what values of these fluxes can give high flux through my objective. The objective function can be biomass or ATP synthesis, okay, and these objective functions are a linear combination of all the other different fluxes that exist within the network. Okay, what we did was we took the human metabolic reconstruction and coupled it with transcriptomic data, some bulk and some single cell, all coming from different experiments, and somehow made this choroidal endothelial cell model used for the production of biomass and collagen synthesis, both which we discussed before, important for migration and proliferation. we confirmed that the model is producing all the known pathways that were reported in literature, like these which are mentioned here. And we ran a bunch of flux-based analysis. I'm not going into the detail of every analysis, but the idea is that we wanted reactions that had narrow flux ranges, which were not changing in any different condition. And we found, and also biomass collagen synthesis, which reactions are essential, and also that are rate limiting for these productions. We found that out. In the end, we focused on SQLA and ALDH18A1, for which there is no literature available in endothelial cells. We did some experimental analysis. We knocked down these genes in vitro, and we found out that as compared to the control, the the proliferation and migration actually reduces in the knockdown endothelial cells. We also confirmed this with in vivo experiments. So basically what we did was we took the mice, the control mice were the ones which had laser-induced injury, and the test mice were actually the same mice which had injections of siRNAs. siRNAs basically reduce the expression of different of these two genes, and thereby we compared them and we saw that, okay, this blood vessel area actually reduces in both the knockdown mice as compared to the control mice. So overall what we understood is that the model actually predicted some new pathways, and we focused on another molecule. I will just quickly tell this because we are out of time. What you can see is that all of these pathways are linked by one reaction, this transporter reaction, SLC25A13. And this was very interesting that only one transporter reaction is at the center. Can we exploit this? Can we try to understand more about this? And this SLC25A13 is involved in malate aspartate shuttle, some biochemistry, but it helps in maintaining redox balance in the cell. But our hypothesis was that it is essential for biomass synthesis. We ran the same experiments and we saw again knockdown of that gene actually reduces proliferation and migration, both which are angiogenic phenotypes. We also additionally what we did was, in the previous study we did not actually verify whether metabolically they are associated or not. So we did some 13C isotope tracer experiments, which means that you tag the carbon of an input source like glutamine, for example, and you trace within the pathway where this molecule is being used. Okay, and thereby we found out that TCA, amino acids, all these go down. Okay, but what does this mean? What does this carbon label fraction exactly mean? So let's say I overlay all of this on this pathway. We see that blocking SLC25A13 reduces everything. So it's kind of, our model is actually explaining something that is really occurring. We can block SLC25A13 and we reduce all the other fractions of metabolites. This also kind of indicates that SLC25A13 has a role linked with biomass synthesis. Okay, just to conclude, metabolism is a complex system, and systems biology approaches can definitely try to dissect this. It's a very painful process to use flux balance analysis or any flux-based analysis on these big models, because you have to set constraints, and it's a big pain. But if you are able to do that, if you invest time in understanding metabolism, you can get some good results that can be experimentally validated. We also saw single cell data was able to identify these cell types, and the cell specific gems could actually identify some target enzymes. Okay, I would like to thank my postdoc supervisor, who I'm collaborating with, continuing with the SLC25A13 story, and all the first authors of the paper. This is our lab. We recently performed Ganesh Chaturthi celebrations. I'm a Punekar, so I wanted to invoke this culture in my lab also. And we did Ganesh Chaturthi celebration. So we are still growing. And if you want to join us, please email me at this email ID. Thank you very much. If there are any questions, I can take it.
Thank you, Abhishek.
Yeah.
Sorry to push you, but we still have time for questions. So
No, it's okay.
if there are any questions, can we have them? Yes, please.
So when you showed your single cell experiment, there was a clear difference in terms of specific pathways being up and so on. So if you look at all those genes and then overlay on top of your, like within that are the enzymes also there, which, so can that be used to parameterize your KEGG pathway? Like you not only say that these are my metabolites and these are at steady state I'm solving them, but now I'm also saying from single cell data that these are actually the enzymes which are actually present given my disease versus control, and will that give us separate information?
So overlaying is possible. You can overlay anything to the network, okay? But does it have a meaning? That is what we need to understand. First of all, with respect to single cell data, the data is very sparse. What I mean by that is, let's say if I take a single cell and I see how many genes I am able to detect after this sequencing experiment, it will be roughly around 3,000 to 4,000, whereas the original number of proteins even that exist in human cell is around 20,000. So we are grossly underestimating what RNAs are actually present within one single cell. Techniques are improving, but it's almost impossible to find out those small changes that are happening within the cell. To answer your second question, continuing to answer the same question, that if you want to overlay those genes, the number of genes are very less. And even if you overlay, you will understand that there are so many connections that underlie the same number of genes. It's not that all of them occur in one single pathway. Okay, like for example, the gene set enrichment analysis we did was using some gene sets that are curated by, let's say, some particular person. The definition of pathway itself actually is very plastic in biology, in my opinion, because a pathway essentially starts from your input and ends to, let's say, biomass or some kind of cellular objective. So it's not that this is glycolysis. This is all work together. That's how I imagine metabolism. So to answer your question, I don't think that overlaying a pathway will give you any useful information about metabolism. Okay.
All right. I think is there a question back? But there is one here. Okay. I think she has been waiting for a while.
Students first, yeah.
Yeah.
Students first.
Yeah. I think let's have the students first. Come on. Let's have that. You actually get one bonus.
Hi, it was a nice talk.
Thanks.
So my question was, when you were performing the experiments on the rats, you were artificially inducing the disease in the rats, right?
Mice.
Mice, and then you took the eye out. And how exactly did you measure the cytokine concentration in the eye that you took out from the mice?
To be honest, that was not done by me. That was done by my colleagues. So I don't know the specifics of how they measured it.
Right.
But as far as I understand, it's they always do a fax to check for markers, and based on markers they will see whether the enrichment. So basically what we did was, actually I'll quickly go to that slide. So we did not characterize the entire tissue. I actually had to skip because of time, but we specifically chose endothelial cells, if you can see here. So we did CD45 minus depletion and CD45 and CD31 depletion. So basically you choose for these markers and select the endothelial cells. So same way, they might have measured cytokine concentrations. I am not sure if that was what was done.
So my question is, as far as I know, wet AMD is a disease that affects the retinal region of the eye.
Yes.
So even if you're dosing in the vitreous humor, you don't know how much of your drug is actually getting into the retina. That's something that we don't know.
Absolutely right. And that is why this choroidal neovascularization model was also questioned by our reviewers. That why did you choose that model? Unfortunately, we found out that, at least through literature search, that CNV is the only way in which you can test for leaky blood vessels. And other retinal development models, they actually just focus on the retina, and you actually cannot take care of the choroid. So CNV was the only way in which you can analyze that. It's a very dirty model, but unfortunately that was what was available. So I'm sure that in years to come, better mice models will develop.
Yeah. I think that's the case in all animal models. Like you will see suppression in vitreous, you will see
Yes.
suppression in your plasma, but
Exactly.
you don't know what's happening at the site of action.
Exactly. And if actually I also rushed through how I made the model actually because of the time limitations. But the model was made for humans and not mice. So we used some single cell data that we had for a human eye actually. A patient was gracious enough to lend his eye. And then we could get some details and we could add it to the GEM model and thereby predict some things. What we tried to correlate between these two analyses is that whatever we predicted from the GEM model was also shown from the single cell data. So they were independent analysis. Okay, but anyway, I think I'm not answering your question, but what I'm trying to say is that you require multiple approaches so that you can validate a particular phenomenon.
But I really liked one point that you said.
Can we just curtail the questions because we need to rush off now, please?
Yes.
You have completed your bonus actually. But still, you want to?
Thanks a lot.
Okay, fine, because we are, we can. You want quickly?
So I just wanted
Yeah.
to, since you stopped in interest of time, could you elaborate a little bit about how you built that model and what methods did you use to integrate the data?
Yeah, okay, sure.
And maybe that can clarify some of the other
Sure,
questions as well.
So basically we started with this Recon 1 and Recon 1 had a lot of problems with respect to gaps and all. So we had to make a complete model out of it. And then we formulated an EC specific biomass function. And we used this refined model and used some activation specific. So basically when you put endothelial cells in culture, they get activated. At that point, whatever transcriptome was collected, that was integrated with the model. For that, I used this.
What method did you use?
For that, we used GIMI, and we specified a threshold using ZFPKM values, and we say that whatever is ZFPKM less than -3 is what we want to eliminate from the model, or rather minimize the fluxes. Based on that, we end up with the base model of endothelial cells. And then we wanted to integrate other transcriptomic data, right? For that, what we did was we did not rely on any other transcriptomic algorithm that can change the bounds. Rather, we said we'll fix the bounds as it is. We will just try to eliminate reactions that have that are downregulated in transcription and they have no effect on biomass. So basically, we just eliminate those reactions. A reason why we did this was because single cell data is so sparse that it could be possible that a transcript was expressed, but it was not captured by the technique. So to account for that, what we did was we just eliminated those reactions which did not affect the biomass. It was also not present in the transcriptomic data and it also did not affect biomass.
And then in
hope
your
I answered
constraints-based
Yeah.
analysis that you did, how did you move from the 32 or the 4 to those 2 targets that you eventually tested?
Good question. So that also I had to skip. So all the 30 targets, we looked for literature search. Like we did a literature survey and we found out that only these 2 genes have actually not been studied in any cell type with respect to biomass and collagen synthesis. So we focused on these genes and then we explored further. I mean, it's just a matter of novelty, right? I mean,
So more of curation, literature curation.
Yes, nothing, no other prioritization. Yeah.
Perfect. Thank you.
Yeah, sure.
Great talk.
Yeah, thank you.
Let's have one more round of applause.